Welcome to the next session for today. We're going to start off with Dr. Aimee Kao. She's an associate professor of the Department of Neurology at UCSF. Aimee?
Great. Thank you, Kathy. I guess I just want to start by saying that, when we heard the news about Paul Allen on Monday, obviously it was so sad. He's way, way, way too young. And I know it's been a hard week for all the folks at the Institute. So in addition to sort of our condolences, we just want to thank you, Kathy, and everyone else for hosting us so graciously. So it's been really sad. So I'm up here representing our team, including Matt Jacobson, who's here. And I'm going to have to skedaddle right after the talk. So if you have questions, you're going to have to ask Matt, as well as Diane Barber and Torsten Wittmann. We were part of this Alzheimer's cohort. The RFA was a call for really new ideas in neurodegeneration, kind of the crazier the better. And at one point, I think Kathy or Tom was quoted as saying we were the craziest of the crazies. So our crazy idea was to think about pH dynamics in Alzheimer's disease. And I will tell you that compared to where we were when we started, we feel like we've made a lot of progress in terms of insight and how we think about this problem. And in fact, it's led us to the point where we're now thinking about selective neuronal vulnerability in a very different way. We think it may actually emanate from a lysosome, the lysosome. And I will define all of those things as we go along. Okay, so I think full disclosure, we think of neurodegenerative diseases as age-related failures of protein homeostasis. And the process of protein homeostasis encompasses the entire life of protein. So when it's made in a properly folded state, then it can be misfolded and then refolded, or if it's not needed or damaged terminally, it can be degraded by many routes. Most people think primarily the proteasome and the lysosome. We know through work in the aging field as well as in neurodegeneration that age, stress, and gene mutations can actually promote protein misfolding. And when we're young, the process of degradation is intact. But with age, there's some data that suggests that some of our protein degradation mechanisms decline. And at that point, the proteins that can't be degraded are sequestered. And so these sort of pathognomonic features of Alzheimer's, Parkinson's, FTD, ALS are seen in neurons. Our bias is that although these aggregates and inclusions are important for defining the diseases, that really they're protective. And this is work from Steve Finkbeiner from over a decade ago, as well as more recently Brad Hyman and others, that suggests that these aggregates are actually protective. Okay, but they're still really interesting in the sense that there's a different set of protein inclusions for each of these diseases listed here, and the diseases actually start in very distinct and stereotyped places. So you don't see Alzheimer's disease typically starting in the frontal lobes, like in frontotemporal lobar degeneration. And so this is this question of how do these diseases achieve selective neuronal vulnerability? And for a long time, it was thought that it was something to do with the anatomy of the neurons, and then more recently, as we began to recognize these autosomal dominant gene mutations, that it had to do with the proteins. There's a problem with that argument, the proteins in which there are mutations that aggregate. There's two problems with that idea, though. One is that these proteins are found in all neurons and sometimes in all cells. And then in addition, there are other gene mutations that lead to these aggregates that are not found in the inclusions, okay? So our approach was to think about some upstream factors, upstream of the sequestration, these protective aggregates that could be contributing to neurodegenerative disease. And so then the question is, how does pH come into all this? Well, as Rick Horvitz said so beautifully this morning, turns out that all cellular functions are compartmentalized and they're dynamic. And one of the features of the different compartments of the cell are that they all have slightly different pHs. And those pHs help to define the compartments, and they're integral to proper functioning of those compartments. And there's a dramatic difference. A mitochondria have a pH of about 8, and then the lysosome actually is the most acidic organelle in the cell at a pH below 5. And each compartment achieves its pH in different ways. We're just starting to understand how the lysosome does this. And in addition to the VATPAs pumping protons in, there are a series of channels and transporters and antiporters that help the lysosome achieve and maintain this acidic pH. And we're working now with Michael Grabe, who's helped to develop mathematical models of how the lysosome achieves acidic pH, and it includes things like the buffering capacity, the membrane potential, the size of the organelles. And so we're really still in the early parts of understanding how this is done, but because of work that was launched with this Allen grant, we're making forward progress on that. And the pH of the lysosome is super important because there are about 60 acid hydrolases in the lysosome that all require an acidic pH in order to function. And among those 60, there are 3 classes of proteases known as cathepsins, and they're defined by the active residue in their cleavage sites as either cysteine, serine, or aspartyl. And it'll become clear later why I'm pointing out these 3 different classes of proteases. Okay, so what led us to think about actually pH in disease? Well, Matt and Diane had actually worked together thinking about pH in cancer cells. And it had been known for years that in cancer cells, the intracellular and extracellular pH were reversed. And what Matt and Diane showed is that if you think about proteination as a post-translational modification like phosphorylation or acetylation, it actually makes a lot of sense that you can actually change the form and function of the modulatable, proteinatable proteins. And that that the pH was very important for metastases, self-proliferation, et cetera, cytoskeletal remodeling. In addition, in 2012, Dan Gottschling had shown that with yeast replicative aging in the mother cell, that the vacuole, the yeast version of the lysosome, becomes progressively more alkaline or less acidic, and that if you can maintain acidity, you can actually extend the replicative lifespan of yeast. And then there's tons of data in neurodegeneration that a lot of disease proteins actually affect lysosome function. And at least a few of them, including presenilin 1, was thought to actually interact with the VATPAs and prevent proper acidification of the lysosome. So this was sort of the state of things when we started thinking about these problems. Now, we sort of wrote our proposal, we read the literature, there are all these tools to measure lysosomal pH, et cetera. But when we actually got down to doing this, we realized that the tools did not, they just weren't up to snuff. And what we had to do is actually develop our own lysosomal pH sensor, which sounds trivial, but it wasn't. And we actually went through half a dozen or more iterations of this using all different fluorophores, targeting sequences. And ultimately, we've come up with one that works great. So it is correctly targeted to the lysosome. It doesn't degrade. It doesn't aggregate in the lysosome, and importantly, the pKa is appropriate for the very acidic pH of the lysosome. So we now have this, we've expressed it in many different kinds of cells. These are HEK293s, we've used MEFs, we've used SHSY5Y cells. We now have this packaged in a virus, so we're getting ready to put them in iPS cells and iPS-derived neurons. And we've been able to move forward to use this tool in a couple different ways. One, we had to validate it, and we had to show that it actually has a pretty linear measurement range between pH 3 and 5, which is perfect for the lysosome. And then we have just put it into C. elegans and shown that it is actually functional. If we treat with a lysosomal inhibitor, baflomycin, which inhibits the VATPAs, you can see that we see a change in the lysosomal pH set point. This is going to be really important for us because one of our hypotheses is that lysosomal pH may be regulated differently in different tissue types and may actually change with age. And so this is an area where right now we're putting the lysosomal pH into different tissues, so neurons, muscle, and intestine, and we'll be able to compare, and we'll be able to do this in live animals with and without stress, with and without neurodegenerative disease proteins. So that's exciting work that is accelerating. So there were a couple different places we wanted to look at lysosomal pH. And the one I'm going to talk about today has to do with this protein known as progranulin, because it's sort of something that we actually know, we've generated a lot of other knowledge that is complementary. So progranulin is actually a mutation. It's a loss of function mutation, though. So haploinsufficiency of progranulin leads to a type of neurodegeneration known as frontotemporal lobar degeneration. In particular, this develops exclusively inclusions with TDP43. And then the levels of progranulin are actually pretty important. If you have too much progranulin, it promotes cancer growth and metastases. And if you don't have any, you actually get a lysosomal storage disease known as neuronal ceroid lipofuscinosis that actually presents early in childhood. So just recently, we took our pH sensor that's encoded in lentivirus and infected SHSY5Y cells that were either control or had been CRISPR-Cas9 edited to be progranulin heterozygotes or progranulin nulls. And what we found is actually kind of interesting. What it seems like is happening is that there's a population of lysosomes that stay just like the controls. And then the lysosomes become just more noisy in the progranulin heterozygotes. We see the higher the fluorescence ratio, the more alkaline the pH. So we're seeing lysosomes that are approaching pH 5, 6, and even higher. And although only one of the two lines reach significance, we definitely see this interesting trend. So we're going to go back and do this in terminally differentiated cells. We tend to see that in cycling cells versus terminally differentiated cells that many things, not just lysosomal pH, but many characteristics of the cells are quite different. So we're going to be doing that. However, it does lead to this question of, what is progranulin doing in the lysosome? So just a little bit more so you understand about progranulin. So progranulin is, it's actually a glycosylated and secreted protein. And I already told you that mutations, haploinsufficiency, lead to neurodegeneration. Interestingly, it's actually a proprotein that can be cleaved into these bioactive peptides known as granulins. And granulins are super, super interesting. They are also maddening in terms of working with them because, as you can see, they're actually incredibly disulfide bonded. Every single granulin, there are 7.5 of them, has 6 disulfide bonds with these 12 cysteines. And they're also sort of tunable by pH because cysteines can be, disulfide bonds can be reduced with protons. And then they also have this very interesting invariant histidine, which is the other protonatable amino acid at sort of physiological pHs. So we started thinking about progranulin and these cleavage fragments. And in data that I have shown in past meetings, and I just don't have time to go over today, we've learned a couple of things about progranulin and granulins. One is that progranulin is actually cleaved into granulins by cysteine proteases. We've sort of done a very sort of exhaustive in vitro protease assays and shown that it's really primarily cysteine proteases that cleave progranulin into granulin. We've also shown in C. elegans that increasing age, as well as certain types of physiological stress, increase granulins. And as I've alluded to with the lysosomal pH, we've shown in C. elegans with a number of behavioral assays that if you remove the granulins and just express granulins, if you remove the progranulin and just express the granulins, you overall decrease animal fitness. So they develop slowly, they can't learn, they tend to have poor reproductive fitness. And now we've sort of localized this decreased fitness to lysosome function. And, this correlates, as Marcus was saying, there's not causal, but we see a correlative increase in lysosomal pH in progranulin mutants. And I should say, when we measure relative progranulin and granulin with age, there is more granulin than progranulin. So this balance of progranulin and granulin seems to be very important. So we're working sort of with this model. So the question is, how are you getting selective age-associated protein accumulation? So to do that, to try and get at that, we just used granulin as a bait and did unbiased IP mass spec and found a list of proteins that the granulins interact with. And there were several protein disulfide isomerases that probably help to fold granulins into their proper state. And interestingly, we found an aspartoprotease that I'm going to tell you more about, as well as VHA12, which is one of the subunits of the VATPase. This is intriguing. We haven't actually had a chance to do much work on that, but I'm going to tell you about what we've learned about this aspartoproteases. So the aspartoprotease in C. elegans is ASP3. The closest ortholog in mammals is cathepsin D. And cathepsin D is a lysosomal protease. It's in that asparto class. There's only 2 aspartoproteases, cathepsin D and cathepsin E. And it has been shown to increase under conditions of stress, starvation, aging. There's some genetic variants that are associated with Alzheimer's disease. It's required for certain processes in neurons, including necrotic cell death. And I had a figure of this, but it actually looked corrupted. The activity of cathepsin D depends both on pH as well as the particular substrate or particular client for that protease. Okay, so we wanted to see if mammalian granulins could also pull down cathepsin D. And Victoria Butler in my lab did a ton of work along with William in Matt's lab. And to summarize, 6 months of work with one sentence, they showed that granulins can interact with, mammalian granulins can interact with cathepsin D, but only, it's seemingly, with the immature forms of cathepsin D, and it seems to interact with the propeptide. The propeptide is sort of a little lid or cap on the protease that prevents the protease from becoming aberrantly activated prior to reaching its proper location. And I should say that they've looked at cysteine proteases, and granulins don't associate with cysteine proteases at all. So the proteases that cleave progranulin into granulin are not the eventual targets of the granulins, and they probably do interact with serine proteases. Okay, so what's the nature of this interaction? So using a set of behavioral assays in C. elegans, we were able to show that in the presence of granulins, we see about a 20 to 25% decrease in cathepsin D activity. That in the presence of granulins, the necrotic cell death is delayed, and this is a process that requires ASP3 cathepsin D activity. And then with our neighbor next door, Bill Seeley, in his brain bank, we actually looked into human progranulin mutation carriers into the autopsy tissue. And compared to controls, where there's no difference between diseased and undiseased regions, the cathepsin D activity in progranulin mutation carriers is much lower in diseased regions. So there's sort of an internal control here. And then this was sort of an off-the-cuff experiment that has really helped us sort of conceptualize granulins and selective vulnerability. We just decided to buy some TDP-43, and we exposed it to different proteases, and you can buy nearly all of these proteases. And it turns out that the aspartoprotease, cathepsin D, is much better at cleaving up TDP-43 than the cysteine proteases that we tested, which doesn't cleave TDP-43 very well. It made us think that actually, perhaps it's a decision that the lysosomes can make in terms of the substrate that it can or cannot cleave under certain conditions that may be leading to TDP-43 accumulating with disease. And then we have a whole other story about tau and selective neuronal vulnerability that I just don't have time to go through today. So here's our working model, and there are still pieces of this that we need to fortify. So progranulin is cleaved into granulins by cysteine proteases. This increases with age and stress. Granulins inhibit lysosomal function or modulate it, negatively modulate it, specifically by interfering with the activity, maturation and activity of cathepsin D ASP3. This protease is particularly important for TDP-43 degradation. When you lose this activity, then TDP-43 accumulates and is forced into these protective inclusions. We have other data that progranulin, the full-length protein, actually can promote cathepsin D activity. So this, this is happening if in haploinsufficiency. You both lose an activator and you gain potentially with age an inhibitor of these proteases. And this leads us to think about different therapeutic approaches, which would include potentially treatments to prevent progranulin cleavage into granulins. Now, this is work that we're actually super excited about. We have this lysosomal pH sensor, though, and one of the things we thought we should do is to start looking at ways that we can alkalinize lysosomes with the hope that a more alkaline lysosome will recruit more proteases and enhance activity. And so really, as a proof of concept, we've converted the assay into these 384 well plates, and we've completed a screen of FDA-approved compounds. This is the validation of the sensitivity of the sensor. And of the 20 or so hits we initially got, one actually survived into our secondary screens called Tillerone. It's actually really an interesting molecule that's probably too toxic to be taken chronically, but it may give us some hints in terms of the way it acts as a proton sink for how we can modulate lysosomal pH. And so with that, I just want to thank the Allen Foundation again. It's been a joy to work with my compatriots. This is our whole team. And we look forward to hopefully bringing some of these ideas to fruition. And I'm an optimist, but as people have been saying, I've been hearing people saying over the last 5 to 10 years that we're going to solve neurodegeneration in 5 to 10 years, and I've never really believed them. But I actually do feel like in the next 5 to 10 years we could make significant headway into different forms of neurodegenerative disease. So thank you very much.
We have time for maybe one question. Mark? There's a microphone coming your way.
So is this a pathway that you're thinking about specific for neural regeneration, or is it a general aging pathway that you would expect to find in other tissues?
I guess I actually think of it as a more general aging pathway. Amber Ney at Stanford actually did a really incredible QTL analysis in killifish and showed that there's a region of chromosome 11 or 15 in killifish that correlates with lifespan. And progranulin was actually right there in the middle of those genes. There were about 20 genes there, and there were several potential lifespan genes. But we have those SNPs. They all either change histidines or cysteine residues. And so we're actually making those SNPs in C. elegans now to see if there's actually an effect on lifespan. I don't know. I mean, you could say that neurodegeneration is simply accelerated aging or slightly aberrant aging. I think there's a gray area there.
Thank you.